All right, it's my pleasure to introduce Xiao-Fei Kong, who's completed his training in medicine. This is unbelievable. The two times that I came to introduce somebody, phone call came through my phone exactly at this very moment. It must be fate. Anyway, so Xiao-Fei completed his medical training at Yangzhou University, and early in his career had a passion for understanding mechanisms of disease, and went from China to France to work with Jean-Laurent Casanova, who at the time was at the Pierre and Marie Curie University. Jean-Laurent is a very well-known geneticist who studies the genetic basis for particularly innate immune defense, looking at Mendelian traits that affect resistance to infection. So Xiao-Fei obtained his PhD in France, and then when Jean-Laurent got recruited to Rockefeller, Xiao-Fei came with him as a postdoctoral fellow, completed his postdoctoral training with him, then went to Mount Sinai, and thereafter to Columbia to do his training in internal medicine and gastroenterology. So as you can imagine, his focus is on genetics of, in this case, the immune system's architecture that controls inflammation in the gut. Some of his seminal work has to do with resistance to mycobacterial infections. So by this point in time, Xiao-Fei has more than 40 papers, one of his most influential ones in Nature Immunology. And he's focusing the beginning of his lab on understanding the genetic determinants that control the emergence of celiac disease. A lot of us have the HLA risk alleles for celiac disease, but only a few of us will get the disease. So that's the mystery that Xiao-Fei is trying to unravel. So Xiao-Fei, we look forward to hearing from you.
And thank you, Ezra, for kind introduction. It's really my great pleasure to be here and introduce our research focus on the human genetics of gastrointestinal inflammation. So as a gastroenterologist, we do a lot of scope, upper endoscopy or colonoscopy. We saw different phenotypes. You can see here, this is candida esophagitis, celiac disease. All the colon have inflammatory bowel disease. As trained in human genetics, my lab focused on understanding the relationship between phenotypes and genotypes, whether such sort of inflammation or infection have a genetic basis. So my lab enrolling patients who present with rare disease, either early onset or severe. They have GI or liver manifestations. Through whole genome and whole exome sequencing, we're hoping to find if they have rare and uncommon, unrecognized genetic disease. And we hope to understand what's the molecular mechanism to determine their clinical phenotype. And through this reverse genetics, hoping to understand to improve the future treatment for the GI disease. And my work was, when I was in New York, I started to study genetic disease, Down syndrome. That's patient with trisomy 21. They have multiple clinical phenotypes. They have intellectual disability, cardiac problem, but they also have a lot of autoimmune disorders. For instance, alopecia, thyroid problem, type 1 diabetes. When I became a GI fellow in New York, I started to more interested in the celiac disease. As this audience have both basic scientists and clinical physician scientists, I want to give you a brief introduction. What is the celiac disease? Celiac disease as an immune-mediated gastrointestinal disease triggered by ingested gluten, and patients usually have chronic diarrhea. They might have nausea, vomiting when they're exposed to the gluten, and iron deficiency, anemia, malnutrition, and osteoporosis are common. They're usually diagnosed at the age of 40 to 60 years old. As Ezra mentioned about, this disease is HLA dependent. They're required for developing celiac disease. It affects 1% of US population. Quality of life can be compromised, and they have the risk of developing lymphoma. So far, there's only one treatment as the gluten-free diet, which is difficult and not always effective for this problem. Celiac diseases mainly affect small bowel. This is the normal structure of the small bowel. You can see the villus. For those with celiac disease, they have villus atrophy, which show here. They have crypt hyperplasia. Crypt is like stem cells in the GI tract. And they also have increased intraepithelial lymphocytes. So when I realized Down syndrome patient prone to celiac disease, a question was asked, what are the risk factors? What are the genetic risk factors for the celiac disease? As I mentioned, in general population is 1%. For those carry HLA DQ2 or DQ8, which are very common allele, 33% of the population carry those alleles, but only 3% of them develop celiac disease. But when we look at the family history, if one celiac index case was found, to looking for the first degree family members, 10 to 12% of their first degree family members also have celiac disease. Another important genetic study was from the TRAIN study. The concordance rate between those dizygotic train or monozygotic train, you can see those monozygotic train, even they have similar environment factors, their concordance rate are much higher, 50 to 70% have the celiac disease. So this is really suggesting there's a genetic factor causing celiac disease. And another interesting finding was from a more rare in-bone error of immunity that's called gain of function stage 3. Those patients have early onset type 1 diabetes, and 20 to 50% of them have celiac disease or celiac enteropathy. So the key question of my research lab, there's a lot of GWAS study in the past to study genome-wide association. Was there be any familial or rare genetic form of the celiac disease was not discovered before? So we conducted a pilot study based on what we know about familial celiac disease to enroll the different families with multiple affected individuals. So you can see this is the pilot enrollment of 9 families, a total of 25 affected individuals. You can see some of the families have 5 even affected individuals. Unlike general population, those families, they have very early onset. Some families have 14 to 12 years old. So, and you can see from this pedigree, it could be some unrecognized dominant trait with a dominant disease have potentially changing our understanding of the celiac disease. So we conducted whole exome sequencing on this on those families. The first thing we looked at is the HLA genotyping. Through the whole exome sequencing, we did fine mapping. You can see almost all the affected individuals carry at least one of the risk alleles causing celiac disease. And you can see actually there's a lot of patients carry compound heterozygous or homozygous for the risk allele. So could it be rare genetic disorders from these 9 families? We conducted different analysis using gene burden test, linkage analysis, and recently we find there's one interesting family. These are 3 affected individuals. They all carry an ultra-rare nonsense variant in a gene called ABCF1. Mom is the carrier. And this variant is not found in any public database. Could this nonsense heterozygous variants explain their phenotype? So we looked at what's going on with this gene. This is actually an interesting gene controlling the cytosolic nucleic acid sensing, and it's regulate CXCR10 and interferon beta production. It's inducible by type 1 interferon and the TNF alpha. And there's also a mouse model on the homozygous deletion of this gene, embryonic lethal. And those mice have heterozygous variants, have been reported have a haploinsufficiency. This gene has been reported to involved in the RPS reaction and controlling the macrophage differentiation. In the mouse model, the APCF1 haploinsufficiency have increased production of pro-inflammatory cytokines. So long story short, it sounds interesting from mouse model that there's haploinsufficiency in the mouse, and we have 3 affected individuals carry this rare nonsense variants. What ABCF1 haploinsufficiency is a new genetic disorder in human causing celiac disease, and how we prove it. So that would be the key question my lab is currently working on. First, we only have 1 family, 3 affected individuals. So we need to find more families through different genetic efforts. And could this ABCF1 heterozygous variants causing immunodysregulation in human? And whether their gluten memory T cell response are altered due to this mutation? And can the RPS response in the mouse, could that also suggestive in the human? We don't know. We're still looking for answer. But for the genetic part, we have the whole genome sequencing data from all of us, and we analyzed all this is the common variants from the whole genome sequencing data, about 1,500 patients with celiac disease. You can see the peak signal was from HLA-DQ. The ABCF1, it's just in the middle. There was also strong significant associate SNPs are associated to the ABCF1. So now we are using different strategy to understanding its mechanism. So we developed different tools to understanding the immune dysregulation. We used a spectral flow cytometer, which can basically measure 40 different cell surface or intracellular markers altogether. These are the peripheral blood PBMC, or using the biopsy sample to delineate different cells might be affected by those rare genetic disorders. And we're also using another tool to study the gluten memory response. We're trying to using different antigen presenting cells, and establish the human T cell library to screening and to study those rare variants found in the human cohort. So this is my last slide. I would like to thank members in my lab and many collaborators both at UT Southwestern and in New York. And thank you.
All right, great. Do we have any questions? Right here, please.
Xiao-Fei, that was fantastic. Have you ruled out the involvement of pathogen, for example, bacteria or virus in this inducing the disease as well, in combination with those genetic predispositions?
The short answer is no. We haven't. So most of so there was some of the earlier studies suggest some early viral infection might predispose those patients for celiac disease. There was early studies on norovirus, but we haven't. But we do have the serum samples from those patients. We can retrospectively see how many viruses they probably encountered in the past.
Nice talk. Is there a candidate gene on chromosome 21, like a STAT or something that looks like it's overexpressed?
So that's a great question. I didn't get clear. So actually, all the interferon receptor genes are located on the chromosome 21. So that's the beginning 10 years ago to study the gene dosage effect of those interferon receptors. And my earlier work demonstrated that their monocytes have showing elevated ISGs, and they also have immune dysregulation. The T cells prone to be activated. So that's also we are hoping to understanding for the downs how their T cell development, how their antigen specific T cells are involved to predispose them for those different autoimmune disease.
All right, great. Why don't we go ahead and thank Xiao-Fei for a great talk. Very nice.